Today I am taking up the topic law of segregation or inheritance of one gene, monohybrid cross, or it is also known as Mendel's first law. All are one and the same. They represent the same experiment. It is known as monohybrid cross because only one pair of inheritance of one pair of alleles is considered here. That's why it's called monohybrid cross. And here, the inheritance of only one gene is taken into consideration, ignoring other genes. It's called law of segregation. Here, the genes or alleles get separated whenever they get a chance. And they get a chance during gametogenesis. So during fertilization, two different alleles come together, and again during gametogenesis they get separated. This is known as law of segregation. Now let us see how this is happening. So Mendel, for his first experiment, he considered only one pair of traits, that is, the height of a plant. So in pea plant, some grow very tall and some very short. And they breed true. A tall plant produces only a tall plant generation after generation. And similarly, a dwarf plant produces only a dwarf plant generation after generation. That means they are pure breeding. So Mendel selected few tall plants and few dwarf plants, and he allowed them to grow for many generations. In pea plant, self-pollination is the rule. Sorry. So he raised them for many generations, and he confirmed that they are pure breeding tall plants and dwarf plants. He called them as pea generation or parent generation. Then what he did? He carried out a cross-pollination between the pure breeding tall plant and the pure breeding dwarf plant. And he got a progeny. That progeny is called the F1 generation or first filial generation. In the F1 generation, all the plants, they were tall. Not even a single dwarf plant appeared. So, all the F1 progeny are tall, like their tall parent. There is no blending of the traits, no intermediate height plants. All were tall, like their tall parent. Then, he allowed the F1 plants to undergo self-pollination. Then what he got? In the next generation, that is in the F2 generation, he obtained both tall and dwarf plants. The dwarf nature or the character which disappeared in the F1 generation reappeared in F2 generation. Then, in the F2 generation, the tall and dwarf plants always appeared in a fixed ratio, that is 3:1. Then he tried the same experiment with other traits and he obtained the same results. So after repeating the experiment many times with the same trait and with other traits, Mendel came to the conclusion that there is something definite or a rule in the inheritance of the genes or alleles. Then, how to explain these results? He conducted an experiment. So the experimental results has to be analyzed. The intelligence of a scientist lies in analyzing the results and arriving at the right conclusion. And Mendel was a successful scientist. He properly analyzed his results. Now let us see how he analyzed the results. Till then, remember, even chromosomes were not discovered, leave alone the genes or alleles. chromosomes were discovered somewhere 30 years after his findings. Then he assumed or he hypothesized that each character is controlled by two factors. Now we are calling them as genes. So he used the term factor in the place of gene. So according to him, or he thought that each trait, here we have considered two traits, tall and dwarf. Each trait is controlled by two factors. These two factors are present in the parents. But during gametogenesis, what happens is the two factors get separated into different gametes. That means during gametogenesis, the chromosomal number is reduced to half. So the number of genes are also reduced to half. Then the trait that appeared in the F1 generation is known as dominant trait. So here what is the dominant trait? Tall. The trait that is suppressed or hidden in the F1 generation is known as recessive trait. Here the recessive trait is dwarf plant. So he hypothesized that the factors controlling a trait are either dominant or recessive. So how to designate them? Always the dominant trait is taken. Here what is the dominant trait? Tall. So the dominant trait is tall. Then the first letter is taken, that is T. To represent a dominant trait, the capital form of T is written, that is T. So capital T represents the dominant trait. The capital letter is also known as uppercase letter. Then it's a small letter, small t. It's small letter or lowercase letter always represents the recessive trait. So here it is the dominant trait. This is the recessive, or we call them as dominant gene and recessive gene. So capital T represents the dominant gene, small t represents the recessive gene. If both the genes are identical, then that plant is known as homozygous plant. If the genes are different or if the alleles are different, then that plant is known as heterozygous plant. Mendel used in his experiments all pure breeding plants. That means all the plants of the P generation are homozygous, either for tallness or for dwarfness. So he gave an explanation like this. In the P generation, in the pure breeding tall plants, the genes are capital T, capital T. Similarly, in the pure breeding dwarf plant, the genes are small t, small t. That means this is homozygous tall, this is homozygous dwarf. So the parental plants are homozygous tall and homozygous recessive. Then they produce gametes. During gametogenesis, what happens? Meiotic division takes place and the chromosomal number is reduced to half. As a result, what happens? Each factor enters a different gamete. Here also, each factor enters a different gamete. So here, in the case of tall plants, because it is homozygous tall, it produces only one type of gamete. That means all the gametes carry the capital T or the dominant gene T. Here also the same thing. Because these are pure plants, they produce only one type of gamete, and all the gametes carry the same gene or factor, that is small t. So during fertilization, these two gametes come together and form a zygote. That means in the zygote, one dominant gene is present and one recessive gene is present. And this zygote develops into F1 generation plants. So in F1 generation plants, we find both the factors. That means the gene for tallness, the gene for the dwarfness. These plants are known as F1 hybrids. Hybrids means they got both the factors. F1 hybrids. These are heterozygous. Then, in the F1 generation, the dominant trait suppresses the recessive trait. That means the dominant trait will not allow the recessive trait to appear. So as a result, what happens? All these F1 plants are tall, like their tall parent, but they are not pure. They are heterozygous. Then, in pea plant, self-pollination is natural, but cross-pollination can also be carried out. Now let us say a cross-pollination is carried out between two F1 plants in the next generation. So this is one F1 plant, this is another F1 plant. Let us say this plant produces pollen grains. This plant produces pollen grains or male gametes, and this plant produces female gametes or ova. This female plant or this plant produces two types of ova. Half of the ova receive this gene and half of the ova receive this gene. That means 50% of the gametes receive the dominant gene and 50% of the gametes receive the recessive gene. Here also the same thing happens. 50% of the pollen grains receive this gene, that is the dominant gene. 50% of the pollen grains receive the recessive gene or this gene. So in this way, in F1 plants, 50% of the ova receive the dominant gene, 50% of the ova receive the recessive gene, and in the case of pollen grains, 50% receive the dominant gene and 50% receive the recessive gene. Then, during fertilization, any gamete can combine with any other gamete. The chance of probability. So here what happens is, this can combine with this one, or this can combine with this one or this one. This combination can be easily represented through a Punnett square map. This is called Punnett square. Here, graphically, we represent how the possibility of combination of these gametes. So here I have written male gametes on the top, and on the left side I have written female gametes. So these two are male gametes, these two are female gametes. So when this pollen grain fertilizes this ovum, what we get? We get two dominant genes. And these plants are homozygous tall. The plants developed from this combination are homozygous tall. If this pollen grain fertilizes this ovum, what do we get? We get a plant which is heterozygous, but it grows tall because it has one dominant gene. If this pollen grain fertilizes this ovum, what happens? Again, we get one heterozygous plant which is tall. It inherits one dominant gene, one recessive gene, and these plants also grow tall like their F1 parent. If this pollen grain fertilizes this egg or ovum, what happens? We get two recessive genes and it develops into a dwarf plant. So in this way, 4 combinations are possible. 4 types of combination of gametes is possible. So this combination, this results in what happens? It results in production of one homologous tall plant, two heterologous tall plants, and one dwarf plant. One dwarf plant. This is known as genotypic ratio. So here the genotypic ratio is 1:2:1. That means 1 homologous tall, 2 heterologous tall, 1 dwarf. That means we can represent the genes also here. Here it is capital T, capital T, capital T, small t, small t, small t. We can represent this in a different way also. Among these 4 plants, 1/4 are homologous tall, 2/4 are heterologous tall, and 1/4 are dwarf. In another way, we can explain 25% of this combination are homologous tall, 50% of this combination are heterologous tall, and 25% are dwarf plants. So, in the law of segregation or inheritance of a single gene, the genotypic ratio is 1:2:1. Genotypic ratio means the ratio taken on the consideration of the presence of alleles, the types of alleles, presence of the types of alleles. Phenotypic ratio. Phenotypic ratio represents the external appearance. Here, 3/4 are tall plants. 3/4 are tall plants and only 1/4 is a dwarf plant. That means they are in 3:1 ratio. So remember, in monohybrid cross, the phenotypic ratio is 3:1 and the genotypic ratio is 1:2:1. Mendel was not satisfied with this. He still doubted whether his results are true or not. So what he did, he carried out the experiments for another term. That means he self-pollinated the plants of F2 generation. That means these plants. And wanted to study how they inherit the trait. Then, in F3 generation, what he did, he allowed all the tall plants to self-pollinate and observed what happens. When he allowed all the tall plants to self-pollinate, among their progeny, 1/3 produced only or produced pure tall plants. That means generation after generation, 1/3 of these plants produced only tall plants. Then 2/3 of this, 2/3 of these plants produced both tall and dwarf progeny like their F1 parent. That means here 2/3 plants produced tall and dwarf plants in 3:1 ratio like their F1 parent. And these results are similar to these results. So, if you take these two into consideration, 1/3, that means this is capital T, plants, that means these plants produce only pure tall plants, and 2/3 produce both tall and dwarf plants in 3:1 ratio. Because these are hybrids, these are pure. And 1/4 plants of progeny, F2 progeny, produce only dwarf plants. That means this. So in this way, he verified his results through the 3rd generation and concluded that his interpretation is correct. His understanding of inheritance of genes is correct. This is about Mendel's first law. Then, now let us consider what is incomplete dominance. So here in this experiment, the dominance is complete. The tallness is completely dominating over the dwarfness. In F1 generation, no dwarf plants appear, only tall plants appear. And also, there is no blending of the traits. Blending means mixing. No intermediate forms appear here in F1 generation of Mendel's first law. Why? This is because of total dominance or complete dominance. That means the dominant gene completely suppresses the recessive gene. But here, in this case, the dominance is not complete. In incomplete dominance, there is no domination of one gene over the other gene. The best example for studying incomplete dominance is snapdragon or antirrhinum plant. These plants produce two types of flowers. Some plants produce only red flowers and some plants produce only white flowers, and they breed true. So in this plant also, naturally, self-fertilization takes place, and red flower plants produce only red flowers generation after generation. Similarly, the plants producing white flowers produce only white flowers generation after generation. Then, scientists wanted to study what happens if the plants producing red flowers are crossed with plants producing white flowers. So what they did, in the P generation or pediatrial generation, they have selected true breeding or pure red flower plants and pure white flower plants and cross-pollinated them. Because of this cross-pollination, in F1 generation, they obtained hybrid plants, F1 hybrids. These F1 hybrids did not produce red flowers or white flowers. Instead, they produced pink flowers. Pink color is intermediate between white and red. Why it happens, they won't understand. When they analyzed the results, they found that it is nothing but due to incomplete dominance. This plant also follows the Mendel's laws. There is no violation of Mendel's laws. How? It is like this. The F1 plants are inbred or self-pollinated. When the F1 plants are self-pollinated, what happened? In F2 generation, they obtained not only pink flowers, but also red flowers and white flowers. It means the red color and white color which disappeared in F1 generation reappeared in F2 generation along with the pink flowers. And they also appeared in a definite ratio. So when the F1 plants are self-pollinated, in F2 generation, red flower producing plants, pink flower producing plants, and white flower producing plants appeared again, but in a different ratio. That is in 1:2, 2:1 ratio. This 1:2, 2:1 ratio is similar to that of the genotypic ratio of the monohybrid cross, 1:2, 2:1. So this genotypic ratio, this phenotypic ratio is one and the same. Then how to explain this inheritance? If you consider the genes capital R for the plants producing red flowers and the recessive alleles smaller for the white flower producing plants, what happens during gametogenesis? These plants produce gametes with dominant gene and these plants produce gametes with the recessive gene. Or let us say capital R, smaller. We can't say here recessive or dominant. Let us use the words terms gene or gene, capital R, gene smaller. So in F1 generation, what happens? This capital R smaller come together and a F1 hybrid is formed. Here, in F1 hybrid, what happens? Both the alleles are present, but they cannot dominate over each other. As a result, what happens? We get one intermediate trait or intermediate color, that is pink color. Then, during gametogenesis again, what happens in the F1 plants? these two alleles, they get separated into different gametes. And just like here, like this, here also 4 combinations are possible. Because of these 4 possible combinations, what do we get? We get red, pink and white flower producing plants in 1:2, 2:1 ratio. And this is called incomplete dominance. So in this way, in plants, complete dominance and incomplete dominance occurs. Thank you.